So people are coming in. More than 100 online, that's nice. So welcome all to this online immunometabolism series. My name is Jan van den Bossche. I work at Amsterdam UMC, and our group is interested in how metabolism controls macrophages. Today, it's my pleasure to introduce David Ferrick from Agilent. He's their Senior Director, New Market Development. And throughout his career, he's been focusing on developing and commercializing life science tools, such as the Cellular, for example, that I think a lot of you are using in your research. And before his commercial career, Davey was a very successful associate professor in immunology. And today he will show you how metabolism tunes and drives immune cells, their fates and their fitness. And he promised me to share some very interesting new data. So I really look forward to this presentation, David. Questions can be raised via the chat box. So afterwards, we'll have time to discuss and get David's opinion on your questions. So I leave the mic to you, David. Go ahead.
Thank you so much, Jan. I really appreciate the opportunity to speak to this group. It's always great to speak to a group that has a passion for metabolism and energetics. Many of us in the field believe, and I'm a new person to the field. I've only been in metabolism, well, okay, 10 years, I guess, is a reasonable period of time by now. But in the field, really believe it's becoming the new biology. It's not this niche curiosity of biochemical pathways and how things well up inside a cell to cause a particular phenotype or support a particular activity of cells. So it's an amazing period of time to be applying these energetic and metabolic tools to diseases. And that's what we've really seen in the last 15, 20 years. And now it's even going beyond applying these tools to understanding disease to applying these tools to actually intervene in disease. So it's a very exciting time for this particular field. And it's joining all the other fields in terms of the toolbox that people need to do this, I think, very direct work with regards to cell function. I'm sure this is a graph that's very familiar to everyone, but it reminds me to share with you that a lot of what I'll talk about today requires a change in our thinking. Instead of looking for a linchpin or a key different characteristic of a pathogenic process like a tumor cell or an infected cell or a degenerating cell, and finding what that lesion is and then correcting it in a very specific way to essentially cause the disease to fall in on itself, right? Like a house of cards, right? Has been pretty much our approach. And the more acute the disease is, the more it seems to work, the less acute and more chronic, it seems that it's more challenging than a single hit. And so by targeting the immune system, we now are taking advantage of the fact that we have a system that is extremely specific, way more than any drug could ever be. Cells just don't touch each other at one point. It's multiple touch points to figure out, is this the one I need or not? There's memory. No drug really has memory. You stop taking the drug, that's pretty much it. And perhaps most important to us is adaptability. The ability of the system to twist and turn with time, and drugs don't really do that, right? Combination therapy, but you might think, when you think about what the immune system can do in terms of adaptability, it makes combination therapy look pretty pedestrian again. So there's a lot of interest, obviously, and there's a lot of, I think, good logic behind intervening in the immune system. This is, however, has caused a gap. And this is something I've been working on my entire career. It's been a lot of fun. How do we give tools to you, the investigators, to do a better job of continuing to up the ante in terms of how we look at living systems and how we look at living cells and how the things we can learn and again, how we can manipulate the systems, hopefully to the benefit of the host. So my talk today will focus on two basic components of targeting and understanding the immune system. On the left-hand side, And don't get me wrong, I don't have a lot of really good answers yet, but I think I can pose some interesting lines that people might look into, and that's the microenvironment. We now, however, all of us would agree, there are extrinsic factors that go beyond any genome, beyond any transcriptome, beyond any proteome or any cell itself that actually influences the behavior of all those factors of cell biology I just mentioned. And we're now just beginning to appreciate the physical dimensions and the various gradients of oxygen, of nutrients, of cytokines that are extremely important in understanding how we manage disease and intervene at more of a population or tissue level. And on the right hand is continuing to look at how immunometabolism and bioenergetic approaches can help us not just learn, but potentially intervene. I want to start with a movie. And the reason I'm starting with a movie is to really demonstrate how much the microenvironment has information that we just don't normally get in most of the experiments we run on plate readers, flow cytometers, RNA-seq, and other such assays, and even metabolomic types of assays. And that is shown here. So this is just a very straightforward experiment looking at a cell that is given a CAR T target, a CD19 CAR T target. On the left-hand side, you have the negative control. It's not expressing the CAR target. On the right-hand side, you see the cell that will be. And what you see as you watch the movie is you see very rapidly, right now there's no T cells. So in red are the tumor cells, the HEC cells, the surrogate. And then the T cells get dropped in. You'll see this kind of grayish mush after 3 or 4 seconds. And then you'll see what the T cells do. And I appreciate that you've probably seen various versions of this movie in various different ways. And the thing that I want to do is I want to stop this movie at various points and make some points. So initially, we see the tumor cells growing without any T cells. You see that they're pretty much evenly spread, and we know they'll create a confluent layer eventually, right? And however, when we drop in the T cells, these grayish guys, they're unlabeled, coming down, and you can see immediately they start to kill cells very aggressively, tumor cells. But what you'll notice, there's no longer a lawn. There's no longer tumors spread out. They're actually being concentrated. And what we actually see is as real killing begins to occur and we get these foci, or what I call these like shark frenzies, right? The tumor cells are alive and they're actually growing in there. And the distance at which these foci of killing are occurring are actually very particular for the type of cell and how it's armed. Different CAR Ts will do this differently with different spatial resolution and different effectiveness. And in some cases, if it's not effective enough or the tumor has the ability to turn off the T cell, you'll eventually start to see red blobs forming instead of the nice dark blobs, which indicate that the immune system is overcoming these tumors. And so I just wanted to show this to present that just getting a cell to kill and proving in a simple assay that you're killing tumor cells is not the whole story. And I think there's a lot of really good research now that appreciates this and beginning to address those components of this answer. So today we'll be talking about mostly the Seahorse platform. And I'm sure that many of you are probably familiar, but in case there are a few newer people to the game of bioenergetics, I thought I would start with my very oversimplified description of aerobic and glycolytic metabolism. These are the two, obviously, major pathways involved in energy, biosynthetic, and redox potential. Those are its 3 major jobs. And what we've learned about aerobic metabolism is it's extremely stable. In fact, most postmitotic cells utilize this because it's very efficient, very stable, very effective. And it's really, really, really good at maintaining great energy balance and providing whatever's needed for the cell. The glycolytic system tends to be more short-lived. It's not a system that tends to be the stable homeostatic system. It's a system that can respond quickly to demands, biosynthetic for growth, energetic for stress, and other such things. So the glycolytic system, the analogy I always use is if you go out and run, I don't care how in shape you are, you could be the best marathon runner in the world, you start running, you're glycolytic because the system's taken on an immediate demand. That's the best response. But to sustain that, to get across the finish line, you need that aerobic system. And so they actually play together very importantly, but they each bring something different to the table. And that's what we're beginning to learn and appreciate. So the one thought that I want to share with you today is what we've learned that to me has been very fascinating is the ratio, the contribution of energetics. So what you're looking now is at our ATP assay. So we can actually convert the flux of oxygen consumption and the flux of proton production, each reflective of aerobic and glycolytic metabolism. We can convert them pretty easily and quite accurately, as I might add, into ATP units. And so now you can look directly at ATP contribution from each pathway, total and each pathway. And what we're finding is that ratio actually has some potentially very valuable information and may be very specific for different cell states, whether pathogenic or not. So things like proliferation, activation, differentiation, and death. And so one of the things I'm going to share with you is there may be very particular ratios that are very distinctive and enable cells to exist in particular states, and that we can potentially exploit this to learn more and to intervene. So a lot of what I'd call modern day modern day bioenergetics and metabolism really came to us. It's not like obesity, diabetes. There was a lot of recognition of mitochondria in degenerative disorders like neurodegenerative. So there was already, in fact, neuro for a long time appreciated this. Just a lot of their model systems and tools didn't allow them to go much further in that. But really cancer metabolism through Craig Thompson, a number of other individuals really, I think, brought home and brought metabolism directly back to disease study and investigation. And as you all know, we appreciate that a tumor cell, although it may grow at the same rate as a proliferating T cell, they do it very differently. And the difference in the metabolic underpinnings of those two proliferations has a lot of knowledge about a malignant versus a non-malignant process. And we learned a lot from that. And a couple of examples of that that just show this very nice, again, this kind of dichotomy of how these two major energetic pathways, mitochondrial respiration and glycolysis, play in. This is a study by the Pino group at MD Anderson, and they were looking at cancer stem cells. And they had this very nice model of RAS. They could control RAS genetically or through a drug. And when they first got RAS positive tumors, they were kind of the prototypic PET scan positive, highly glycolytic, chewing up glucose that we see commonly in many, many types of cancers. And these are actually quite treatable by actually a lot of conventional therapies as well as targeted. However, when you extinguish the RAS, the tumors go away, and eventually these mice will regress. But what comes back out is now a very different type of tumor. It has much more stem cell-like qualities, and it's very intractable to treatment. And what you see is a very simple phenotype, at least energetically. The RAS tumors have this very high glycolytic flux. The recurring tumor, very low. Essentially, they flipped. So now the relapsed tumor is very aerobic, as you can see here, versus the very glycolytic RAS tumor. So if you actually take and try to say, okay, is there an energetic profile, different categories of malignant energetic programs, let's say, that drive, so you could maybe bucket cancer into these kind of basic groups. We kind of did a very simple experiment in our lab that was kind of fun. So the first thing we did when we had this ATP assay is we took just a whole bunch of NIH, the straight 60. Maybe it would be better to do this in primary tumors, and I know some people are doing that. But what you see, at least in this group of highly utilized tumor lines, is that you get a very diverse profile, pretty much almost any particular level of aerobic, and quite a few different levels. The reason these look scattered is because we lined them up based on their oxygen consumption. So it looks to be pretty random if you looked at it. And we know that this ATP is not just for the phenotype of the malignant growth, but it also includes the culture adaptation. So we would have thought culture adaptation might have, caused it to be a little bit more similar, but basically it's random. However, if you take the ratio of these, the glycolytic ratio to the aerobic, they actually start to cluster into various groups. So if you take the ratio, they start to actually cluster. So this kind of gave us a first clue that there may be particular equilibrium defined by the ratio of glycolytic to aerobic metabolism. Excuse me, I'll take a drink here. That there might be certain equilibrium or set points that are very optimal for certain types of malignant or normal cell function. And this is yet another cancer study showing how the metabolic system can be very uniquely adapted. This was work done by Ralph de Berardinis' lab. It's a beautiful, beautiful study combining metabolomic tracing with using a 3D model of a spheroid analyzed by oxygen consumption by the seahorse. And what was really key about this is he noticed that instead of the oxidative pathway for alpha-ketoglutarate coming in from glutamine, that there was a reductive pathway that actually alpha-ketoglutarate could be used reductively. And why would a tumor cell do this? Well, the first problem he had was proving that tumor cells do these and not normal cells. And it turns out that only in 3D will a tumor cell use reductive carboxylation. So in normal monolayer growth, tumor cells will, in many cases, use glutamine oxidatively. And so what he showed is that 3D, so first showing that 3D formed a relationship, think of the microenvironment that enabled the cells, by virtue of that, to use glutamine reductively. And the reason they were using it reductively, a very novel pathway, he demonstrates. If you are into metabolomics and seahorse, and you want to really see the power of both, this is an amazing read. The way they went about thoughtfully dissecting out and using tracing to really identify which way the flux was moving is pure genius. It's an amazing read. But basically what was happening is the tumor cell created a futile cycle of these glutamine carbons to citrate. It wasn't helping biosynthetically, but what it was doing is grabbing reducing equivalents from the cytosol and bring them into the mitochondria. Because it turns out that in these particular tumors, in many tumors, there's a real liability of redox in the mitochondria. And that can be subverted by bringing those reducing equivalents in. And it turns out that so far, they've never seen this in a normal cell. They've only seen this in tumor cells. So it also represents a potentially nice targeting approach. And you know there are many compounds out now for the IDH1 and 2 genes as well, enzymes as well. So now let's move into immunometabolism and kind of the main story that I want to share with you today. There were there were a few individuals that had been exploring the metabolic principles of immune cells. Craig Beeson, Jeff Rathmel, and a few others, but it was mostly a passion for a few. And I think really Erica Pierce's work was one of the first works that we saw where it really started to utilize these tools to really go after unique properties of the biology of the cells. And she was the first really to very definitively report the importance of mitochondrial biogenesis and mitochondria in the stability and homeostasis of memory cells, allowing them to live very stably and, more importantly, be rewired in their programming to more quickly and effectively react to the return of a pathogenic process. And so we got this first dichotomy, very simple, but I think it holds water in many cases, where cells that have high biosynthetic demands, meaning effector cells that have to proliferate and differentiate and have high need to either produce cytokines or to directly kill cells, take on this aerobic glycolysis. So a combination of aerobic and glycolytic metabolism to meet those needs. However, cells such as these memory cells that are, when they're quiescent at a lower energy level, really rely on a very stable mitochondrial network to not only for longevity, but for reactivation. So she saw this very first distinction. And actually one of the benefits of studying the immune system actually versus neurons or cardiomyocytes or hepatocytes is that you have all these various different populations that do pretty much one thing, right? It's a network of numbers as opposed to one cell you know, evolving to slightly different states of differentiation and function. And so it's become very tractable to very quickly identify unique metabolic signatures and then begin to understand how those metabolic signatures influence the behavior of these cells that have very unique tasks. I think as we began to learn that, we'll be able to migrate that into other cell types and better understand how the metabolic changes in cardiomyocytes and neurons help. So Madhu Sukumar and Rick Nestifo's lab took this idea very quickly and said, okay, the idea here would be, if I actually alter metabolism to make T cells have greater aerobic metabolism, would that alone affect the balance of memory versus effector? And you could see the huge implications for things like vaccines. And of course, with COVID, there's a lot of work being done in understanding that principle. And so the idea was that could I influence, just by altering metabolic behavior, could I influence the balance of an immune response? And so what he did is a proof of concept study that I thought was very nice, using a traditional transfer model. So you transfer immune cells from one animal into another. So presumably you're only transferring the intrinsic protective properties in a particular cell type. And therefore you can see, is it sufficient to provide protective immunity into the host? So this is a very standard model, melanoma model, where you can prime to a melanoma antigen, transfer those cells and see if they're protected to the melanoma tumor. So what he did is when he primed the mice, and so the idea was that if the cells are more aerobic, and using 2-deoxyglucose, you can make cells more aerobic, would you get more memory? And in fact, he also made them more glycolytic. And by making them more glycolytic, could he also make them better effector cells? So he took, and this is the PML, very common model, transgenic model, very easy to work with. And he showed that 3 days of very low doses of 2DG, and this is important, it's not a big toxic dose where everything is blunted. You see that glycolysis as measured by ECAR is reduced in the treated red population. If you take the ratio, they are more aerobic. You transfer these into animals, and you can see that they are significantly protected. So this was the first nice proof of concept that by presumably just altering the metabolic poise, one could influence how the immune system would evolve. And so I think I've made it very clear, but this is a slide that always stops me to make sure that I make my point. And that is that when I learned biochemistry, I hope it's different. Maybe with people like Jan and others, it's very different now. But when I learned it, it was a textbook with these ridiculous pathways. I think it was the German version, much more detailed than the Subway version. And it just occurred to me when I walked away from biochemistry after I took the test and memorized everything was that anything can happen. You can get ATP from 20 sources, you can get nucleotides from 10 different alternative things. And it just seemed it was great because think about it. If you're not alive, if your metabolic system is not in equilibrium, and therefore it's great that you're not reliant on one pathway to get energy, because if that pathway gets hurt, you're kind of messed up, right? What I did not appreciate is that the equilibrium that allows very stable long-term phenotype, like homeostasis, like us being healthy, there are actually only a few good solutions that create equilibrium sufficient or pathology. In other words, there's only a few good energetic solutions that make the cancer so stable that it takes out the host. Think about it. Cancer is extremely stable phenotype. It's one of the most stable phenotypes I know. And when I was taught, we were taught cancer is this crazy, unwild, ridiculous, uncontrolled growth. But actually, I might argue that it's extremely stable. And so what we've learned is that not every solution is possible. So if you look at what I just said and showed data of, in the next few slides, I'm going to try to make the point that if there's only a few good solutions that can support, let's say, memory development, then if we know what that program is, we can maybe engineer it to get the outcome we're looking for, right? So let's talk about 3 different ways that one can begin to use energetics to influence immune function by moving this equilibrium into different states that would only allow certain biological outcomes to occur effectively if we believe that there are only a few good solutions that can keep a cell in a good equilibrium. And so one is immune cell fate. And the Carl June group actually stumbled upon this. This wasn't an intentional thing. But serendipity is a great asset if you're aware in the lab. And that's what I always used to tell my students, and at UMC, I heard it from someone else, don't throw out the experiment that didn't work. Think about it for a while before you start the new one that it goes the way you think it should go. And this is what happened here. So as you all know, story is quite well known, is the June group took a empirical approach, just took a number of different signaling domains in the T cell receptor and said, can we turbocharge? Can we ramp up the effectiveness of a patient's T cell? I don't know how much they were thinking about immune checkpoint at that time, maybe not a lot, but just going in there and doing what we can through a synthetic approach. And it turns out that for blood tumors, the combination of 4-1BB and zeta worked quite well. And I'll show you in a couple slides why that's likely the case. But inadvertently, it turned out CD28, which would have been the front runner by anyone's guess to be the right co-receptor, turned out to not work well in blood tumors. But it actually, with the success of the blood tumors, when they started to move into solid tumors, which is, of course, as you know, the real challenge, it turns out that 4-1BB didn't work very well at all. In fact, almost no CARs work. They're still learning how to make CARs work in solid tumors. But CD28 seemed to work better. So the idea was, well, what was the difference? Long story short, and this is their first XF experiment. So, pretty good. Could do a little optimization, but pretty darn obvious that the 4-1BB had more mitochondrial respiration, therefore potentially a more aerobic program than did the CD28 zeta. And from the immunity paper, it was a very beautiful job of showing exactly what was happening, is they were pushing the metabolic equilibrium towards a more aerobic, in the case of 4-1BB for these T cells, which brought out more mechanisms associated with persistence and central memory, versus the CD28, which seemed to improve effector development and function. And it's not that you need one or the other. And in fact, many people now believe that depending on how a patient presents, maybe their issue is persistence, and maybe aerobic 4-1BB is the way to go. But others, maybe they're not very immunogenic. They're not developing a good enough response, and that response is not persistent enough. And maybe they need aerobic pathway will be more beneficial. And so that's kind of started this line of thinking. But it's not just CD8 T cells or CD4 T cells, Tregs, which has kind of come to the front a little bit in terms. These are the cells that try to establish that balance between reactivity and managing responsiveness. And we know that a lot of times the ill effects you feel from being infected, and again, COVID's a perfect example, have more to do with the immune system than the infectious agent. And the immune reactivity is the one, we have many, many states where immunoreactivity is a significant, if not the component of the pathological effect that is malignant, or dangerous, I should say. So here we show two papers that show that actually Tregs utilize both glycolytic and aerobic, and that sometimes causes a problem because in the tumor microenvironment that's low in glucose in the solid tumor, that is definitely hurts the effector cells that are much more dependent on glucose for their function. But the regs maybe are more resilient because they are using aerobic. But aerobic metabolism is a key feature of their immunosuppressive activity. And so we now know, we have very good evidence of how Tregs can be one of the issues in the microenvironment. And then there's even a newer paper on the right, a real recent paper showing that actually you can begin to split up the two pathways in terms of some specialization, where glycolytic function seems to be very important for migration into various areas versus aerobic, which is clearly required for the immunosuppressive activity. So we're beginning to see how the pathways also have specialization and not just can cooperate with each other to provide the support. And then macrophages, I'm going to go through this quickly because this is one that I'm assuming a lot of people know is, but again, many cell types are fitting into this simple dichotomy, if you will, that I first raised, which is the aerobic versus the glycolytic contribution. And we're seeing that early, immediate demands, relatively short-lived activities tend to really be optimal for the glycolytic system because of its incredible ability to dramatically ramp up and respond immediately under many different conditions. and the aerobic system being one that is very stable, highly integrated, very associated with homeostatic types of mechanisms. And you begin to see this play out in the different cell types, memory cells, M2s being more the aerobic, and M1s, and effector cells, and activated cells being the more glycolytic demanding types of cells. This assay I love. This is one we did in-house that really got us very excited, and a very nice story in China, because we were developing, we thought this would be a great assay for people trying to increase immune reactivity, you know, kind of the immunotherapy. What you're looking at is very classic macrophages that respond well to LPS and low responders. And you can see here that if you give LPS or LPS plus a second signal, gamma interferon, which is of course equally important for full maturation and activity of a macrophage, you can see that they both increased in about 2 to 3 hours here. And we can see the low responder immediately we can detect because of its lack of glycolytic increase, which we know is important for early stages of activation. Now, if we let this experiment go for 6 hours, we see something that's really interesting. We see that gamma interferon has a second glycolytic peak, which kind of makes sense, right? Because it adds alternative capabilities to that macrophage. But what we see, which is really interesting, and we actually worked with Ed Pierce on this quite a bit, was very nice, is that the gamma interferon peak is essentially, the engine is essentially intact in both cells. It's pretty much different. You can tell this very nice and very clearly. It's really, so if you're focused on trying to say, okay, how can I get this cell type to be a better reactive, in this case LPS or pathomimetic, clearly you need to focus on the early activation, right? That's where the difference is. But in the ability to become fully activated by gamma interferon mechanisms, they seem to be relatively the same. And so let's now look at how you can, I've been talking about that there are certain equilibrium of glycolytic and aerobic metabolism that create a stable phenotype or a phenotype of a particular cell. But when you also have a system that can look at activity over time, you can actually see cells transitioning between different bioenergetic states. And so by just watching over time, like I showed you in the macrophage, we can learn a lot about how cells transition between and the different stimuli that can influence those transitions. This is a paper by the Delgoff group that I really like. And what I've done is I pulled out the bioenergetic data. There was a number of other beautiful flow data and other types of information. And again, I'm showing you the bioenergetic data. I'm sure you all appreciate everybody uses more than one tool. You can't build a house with just a hammer, right? You need a saw, hammer, a drill, screwdriver. And that's the same principle in the lab. We have a number of tools we use and we harmonize them to tell a very nice story. What's really neat about this one is I can just pull out the energetic data and show very nicely how they kind of did another look at early T cell activation. So before this study, you know, most people knew about the signaling cascade LCK-ZAP70. People understood how pyruvate dehydrogenase regulates pyruvate moving between the mitochondria and lactate, the aerobic versus the anaerobic pathway. And we knew many different components that were involved in supporting the glycolytic system that supports the proliferation and differentiation and acquisition of effector function in immune cells. And we have players like AKT, HIF-1 alpha, CD28 co-receptor, increase in glucose uptake, on and on. And what he did is he just asked a simple question. If I have a previously activated T cell, does it still play by the same rules? What are the requirements? Has anything changed in an educated T cell in terms of its metabolic poise, or let's say its wiring? And so the first experiment, using glycolysis flux, glycolytic fluxes by acidification rate, is looking at did activation of these previously activated CD8 cells require de novo transcription and translation. And you can see here, the black is just the control activated with CD3, CD28, these CD8s. And you can see that you don't need any de novo synthesis of protein or gene. So you can knock out AKT and HIF-1 alpha as being required to reactivate the CD8 T cells, at least early on. If you then look at CD28 co-receptor activity, you can go out to 10 hours. I think they actually went out to 30 hours total. And again, there was no requirement for CD28 co-stimulation. And so now they're beginning to say, well, what is going on? What is the minimal requirement for reactivation of these cells? And so they use dichloryl acetate, which many of you may know about. Dichloryl acetate does a nice job of influencing pyruvate dehydrogenase kinase 1. And by doing this, it will shunt pyruvate into the aerobic pathway. In fact, it's being used in clinical trials to try to help remove the malignant growth by shunting the anaerobic glycolytic pathway. Supports a lot of tumor growth. And you can see that dichloroacetate completely inhibits this rise in glycolysis. And so it will actually inhibit the activation of these cells. They will not become activated with DCA. And you can see now they had their first clue that it seems like the system simply switches the pyruvate channeling, excuse me, between the mitochondria and the anaerobic system. And that might be a very effective way to immediately get the cells moving in the direction you want. And then the last thing to do was to use injector ports to look at a number of pathway inhibitors to the various pathways that we know of. And sure enough, the only one that was able to inhibit this increase in glycolysis was LCK. And so now you end up with a kind of an interesting, nice, straightforward kind of picture where once the T cell engages the appropriate signals for activation through LCK-ZAP70 to PDHK1, and you simply move pyruvate. I'm sure it's not that simple, but you get a very nice clean switch into this glycolytic system that then can really rapidly put that cell on its journey to activation, proliferation, and effective function. So the last thing I want to talk about here is immune cell fitness. It's a very exciting new area. This is where I have some especially new data to share with this crowd. I'm going to assume in this crowd that is knowledgeable about bioenergetics, and especially those of you who are quite familiar with mitochondria, this will seem very anticlimactic to you because mitochondria, it's very simple. If you have good mitochondria and they're working well, you're going to be pretty fit. If you don't have good mitochondria, you're not going to be very fit. And I think what we're learning is one of the best ways to measure the fitness of a cell may be looking at its mitochondrial and glycolytic pathways. And so what does that look like? So one of the challenges, and I'm not looking here at a bioenergetic assay, you're actually looking at a killing assay, potency assay using another instrument called the Excelligence. And what you're measuring on the y-axis is impedance, which is a great measure of cell proliferation and killing. And so as you go up the Y scale, so you're looking here now, these are just tumors, and you can see over a day, the tumors start growing. They will increase the impedance because they're growing on that plate where the electrical field is. And you can see that signal goes up and it's extremely sensitive. And so what happens at this point is we add our effector cells. So look at the sensitivity of this assay. When I was running killing assays with LDH and chromium, I had to at least have 2-to-1, 5-to-1 to see any effect. This is extremely sensitive assay. You can see at more physiological ratios of effector to target. What you see here is that these are various different, this is Nabil Ahmed who shared this data with us. It's not Mark Liddell's data, he was just making a comment. And what he was doing was trying to find different CAR constructs that would be very effective. I believe this is a glioblastoma model that they were investigating, developing a CAR for. And you can see 3 of those constructs performed quite well. They were very rapid and effective at killing. But if you go out 5 or 6 days, you see that the tumor cells started growing back in 2 of the constructs. So here we have an example where either the CAR started to fail, or in the case we now know, he now knows in 2 of them that the tumor actually is starting to turn off. So remember I showed you that earlier movie. Something is happening that in that early killing, I don't know if you're selecting for certain tumors or if certain tumors can react in a certain way, but they eventually blunt the T cell effectiveness. This is something we're looking for. So what might be involved in this loss, this exhaustion of this immune response? And this brings us to another Delgoff paper where they showed, and they showed very nicely with a flow reagent using this mitotracker deep red FM that they validated as a kind of a nice, kind of qualitative way at looking at relative levels of mitochondrial mass. And he showed very nicely in this study that it was really the tumor microenvironment that was really enforcing a decrease in T cell function by reducing mitochondrial mass, is basically the story here. And you can see very nicely that if you compare CD8 T cells across the non-draining, draining lymph node, which would be presumably draining from the infection or the tumor in this case, the infiltrating cells and the effector cells, this is a control. These are effectors that are not in the tumor microenvironment. You can see that the microenvironment was unique in decreasing immune reactivity, exhausting the cells, and it correlated very highly with loss of mitochondrial function. And he showed very nicely, very simple proof of concept that if you enforce through PGC1 alpha, which we know causes mitochondrial biogenesis, if you enforce and enable the cells to increase mitochondrial activity, they will start making cytokines again, and they will start killing effectively. And he showed in a very nice model, again, by using PGC1 alpha, one could increase the persistence, presumably, and the overall effectiveness of the immune response. So then he did another study. I really like it. It's a kind of a nice progression here. So then the lab said, okay, what if, could I develop kind of a therapeutic regime with the principle coming back all the way to Madhu's work that says, if I increase mitochondrial respiration, period, will that overall help the in vivo host in its ability to respond and overcome a pathogenic insult? And so here's looking at just 24-hour in vitro stimulation of CD8 cells. This is in a mouse model, by the way. You can see, and what's nice here is he used antibodies that are in clinical trials. So these are antibodies that are being used in various clinical trials in combination. So this is anti-CD28, anti-4-1BB. And you can see, as we showed earlier, that 4-1BB will dramatically increase mitochondrial maximal, and really it increases mitochondrial mass and biogenesis. So then he does the mock in vivo. Well, if I treat in vivo with these antibodies, can I see an increase? If I take those CD8 lymph node cells out, can I see the same increase in mitochondrial mass as measured by spare capacity? And yes, that is true. So you have your isotype control. Then you have PD-1, which is actually PD-1 is known to increase aerobic metabolism. You can see it can do that. A PD-1 combination, but 4-1BB by itself was superior in increasing the mitochondrial. So this is a 3-day in vivo treatment of these antibodies, and then looking to see if the mitochondria were increased, and maybe these mice be more protected. So now comes the challenge. So you, once a week for 5 weeks, inject each of these antibodies, PD-1 alone, plus 4-1BB continuous, and then starting with pre-treatment with 4-1BB followed by PD-1. And the reason they did this is 4-1BB in trials is showing toxicity. And so the idea was, could we, with 4-1BB, get the increase in mitochondria, and would it persist for some period of time, enough time to be consistent? And you can see here, it did a very nice job. Again, a proof of concept, just simply increasing mitochondria. Could I somehow alter the ability of the microenvironment or tumor to cause this paralysis of these immune cells? I'm going to skip this one and go right to this one. So this is new unpublished data from Roddy O'Connor in the Carl June group that he shared with me, and he was going to present. In fact, I think there's a virtual ASGCT meeting going, so I'm sure he'll be presenting a much more detailed version of this story. But basically, there was a paper that came out a couple of years ago that showed how high levels of arginine could influence anti-tumor activity, and it was highly correlated with increasing mitochondrial function. And what you see here is an attempt. So many groups are trying to see if a CAR-T approach could work in a solid tumor. And as I told you earlier, it's been quite unsuccessful thus far. So this is a prostate, in vivo prostate model using PC3 tumors in mice, very well established model. They're targeting the CARs. It's the same type of 4-1BB zeta kind of thing, targeting against the prostate tumor antigen. if you grow the cells, engineer and grow them in the normal 1 millimolar arginine versus 4, you can see a dramatically improved in vivo effect. So they're grown in arginine, then they're injected in the mice, and you're now looking at the tumor size in the mice. And what's really interesting about the story is it turns out that if you treat with high levels of arginine up to 7 days and then wash the arginine out. So think about manufacturing a car. You manufacture, you've got some period of time to try to condition cells. And so when you wash them out and go to day 15, you can still see that the cells where the arginine was washed out, the high levels, 4 millimolar high level back down to 1, maintain this increased state. And it turns out they've begun to do metabolomic analysis, and it turns out it seems to be connected with the branched chain amino acids, which is actually a cool story. And it makes sense to a lot of people that study branched chain amino acids and its connection. So there seems to be a connection. It's not arginine metabolism, they believe itself, that is directly influencing the mitochondria, but through the branched chain amino acid pathways, maybe where there's a direct effect on mitochondrial activity. And so the point here is the potential to influence the metabolic poise in such a way to really provide more persistent immunity in vivo. I'm going to skip this, and we'll go to the punchline. So what have we learned? And what is the idea that many are seeing, or the potential opportunity, let's say? This is my version, but I think many people in the field will tell a fairly similar story. I'm trying to do the best I can to kind of collect what I'm hearing as well, but realize biased to the speaker. So what we've learned is that if you look at the continuum, this ratio of these two pathways, we see that naive cells, we now are pretty comfortable with the idea that they're very low energetic because they're quiescent, it's given. They're quite aerobic, and we believe this is what keeps them happy and sedate and ready to be activated. We know that when they're activated, as we showed you, they become glycolytic. They increase both aerobic and glycolytic, but glycolytic metabolism more so, and is required to support the high demand for growth and differentiation and acquisition of effector function. So you can see here by the little bar, it's now highly increased, and you see this contribution of aerobic metabolism. And we know that these cells are very effective at killing or getting rid or producing antibody or producing cytokines if you're a CD4. And we know that these memory cells that go back down have a slightly different profile that is indicative of this education that they received in the war against the pathogen. In immunology, I'm hoping that everyone here watching this presentation is healthy and well today, which means your immune system is working really good. And in fact, many of us, fortunately, will live our lives where this cycle works just like this. Something comes in, perturbs the system figures it out, finds it, and eventually gets rid of it. In fact, you could argue that in a competent immune system, you're healthy. That's, I think, the attraction of immunotherapy. So we might argue that the only time you have a chronic viral or a chronic age-related disorder, pathological through inflammation and obesity, diabetes, whatever that might be, is due to some compromising component, whether intrinsic, I'm too young, or I'm too old. Well, in the case of being too young, that might help you for COVID, actually, as the case may turn out to be, or too old. But then there are extrinsic factors like the tumor microenvironment or the pathogen itself. Viruses do this too, not just tumors, have ways of interfering with this immunity, this network. And so what we've learned is that chronic infection, what I showed with you today, is that maybe the normal way that we turn off T cells through Tregs and other things is we simply reduce their mitochondrial via various mechanisms, we reduce their mitochondria. And isn't that a great way to put a cell to sleep? Just start reducing its mitochondria. Eventually, it's going to lower its activity. Eventually, it will go away through essentially an exhaustion. And what the tumor may be doing is prematurely exhausting these cells by taking advantage of the fact of lowering its mitochondria. Maybe via normal physiological, that they usurp, or maybe some other way to reduce mitochondrial activity. But we also learned that we can intervene. But what's really happening in healthy people, and that's the thing here, is that I was trying to explain earlier, is one thing we do not model yet in any of the work we've been doing, and Jan and I were talking about this before the presentation, we do not model how an immune system actually evolves over the infection and pathogenic insult. And we know, any immunologist knows, that the antigen first recognized is not necessarily the antigen the system eventually overcomes the infection with. There's this affinity maturation that occurs throughout the response, and we're not modeling that quite yet. But maybe in patients that have responded very well and have great durable responses, it's because we've added the one thing that was missing, or the gap that was occurring, and then the system recalibrates as a result of that, and then provides. And so if you think about this oscillation, where the immune system is reacting in numbers, in the environment, to the appropriate signals, that then leads to greater killing. But then at some point, it goes back and forth between these, oscillating between the pathogen getting the upper hand and the lower hand, and eventually it oscillates downward. You're going to end up fully recovering and having protective immunity. If this begins to oscillate upwards, it means the pathogen is getting the upper hand by any number of mechanisms we're beginning to learn. So we need to keep in mind that the immune system is highly adaptive. That's great, but it's not a one-shot deal, right? Although we do appreciate, otherwise no car would have ever worked, that certain people, they just need that hit. And then the question is that sufficient on its own? Most people believe a car on its own in an immunodeficient patient is not going to cure you from the cancer, no matter how effective it is killing the tumor cell. So it's basically getting the system back engaged. And with that, I just like to end with this. I think if you're in the energetic field, you should be very happy now. The last couple of years have provided Nobel laureates that have directly been utilizing the concepts that we've been talking about here today. And I think we're just going to see more of this. It's really bringing back the fundamental way that a cell does its physical tasks is through energetics and metabolic pathways. And the better we are at measuring, the better we are at understanding those equilibriums and how we achieve them, I think the better we'll understand the biology that we've developed all these other markers for that allow us to know what it looks like when it's right. What we don't know is how it gets to there, right? In the US, we always use the cow leaves the barn, right? We don't know how it left the barn. That's what we're trying to figure out. So with that, I appreciate your attention. I'm happy to answer any questions you might have.
Thanks a lot, David. This was very nice and useful. I like this a lot, and especially how you tied together immunology, metabolism, and cancer biology. I think this was useful for a very broad audience. So thanks a lot. There are quite some questions. We're close to an hour, but I think we will address some, and we can, depending on your time also, but we can go over an hour and see where we go.
We'll take a look right now. I'm pretty sure that that's perfectly fine by me.
Okay, good. So I had an initial question. When considering this ECAR and OCR readings in the seahorse, sometimes if you activate an immune cell, can be a T cell, can be a macrophage, you see this hypermetabolic kind of switch showing a very high ECAR and both OCR. And what people then question is, okay, obviously the glucose flux goes up and the increased ECAR suggests there is lactate being produced, but it also shows that glucose is going into the mitochondria and is fueling those. So obviously other things are contributing to this ECAR, and if you correct for the CO2 being produced, that helps. But still, what other factors are contributing to this ECAR, you think?
Right. So let's first start with just the measurement and what we learned about the contributors to protons that get extruded from the cell. And when we did that work, which was part of the ATP assay, but it's also this glycolysis, just GRA rate assay, we tried to really come up with a quantifiable number of protons, picomole per minute, that would reflect those that are coming from that anaerobic process. And you're right, there are sinks that absorb these protons depending on differences as to how the cells are supporting that. And what we realized is you could not take, a couple people like Martin Brand, some of the real wizards in the field, tried to take a stoichiometry, a balanced kind of approach of trying to calculate where these sources could be based on nutrients and a number of other factors that might cause these types of things. And it's just too complicated. It's just too complicated to, we just don't know enough to do those kinds of predictions. We don't have enough control over the system or enough measurements of the various different points at which protons can be absorbed or contribute. There's also the measurement side of it. Because of the cells where they are, the permeability of the plastic, there's a number of really significant, let's say, artificial components of measurement, right, that we all appreciate that can contribute, right? Like protons move into the plastic, oxygen moves into the plastic. And of course, we have algorithms to essentially create coefficients to account for those effects. And so the long story short is we used an empirical approach where we looked at what the rate was at both only aerobic glucose metabolism and blunting it and only having mitochondrial and other activities. Mm-hmm. Kind of made those the two boundaries, if you will. And then we built the coefficients based upon what is actually happening in that particular measurement technology. And when we did that and accounted for those coefficients, it started to equal lactate levels. It's not exact, but it's really close. And we felt that even though that might not reflect perfectly all the protons from glycolysis, it reflects the outcome of the anaerobic pathway quite well. So we could at least account for that. So that's kind of the technical end of it. So what we have now in the ATP and the GRA assay is a better accounting and more corollary between lactate, which is the known, endpoint for the anaerobic pathway, and our measure, right? We know lactate can be handled different ways with transport. So we think if you're following protons, which there was a science paper or a wiki many years ago. And one of the things that we first established this principle on was that he did a very nice job of showing all the various sources of protons within a cell. But anaerobic glycolysis by far was like orders of magnitude above almost all the rest. And that's why, in general, it's at least directionally correct. And what we've done, I think, is make it. So anyways, in our empirical process, the contribution of protons from the mitochondria was by far the second most biggest contributor. Once we eliminated that, we had a measurement that was very close to at least what we see with lactate. Now, on the other side, if part of the other question was, what's happening biologically? I think that's a very good question as well. There are certain situations in biologies where you might anticipate different things going on, right? And so perhaps with time. But I would have to say that thus far with tumor cells, with immune cells, with cardiomyocytes and neuro, where the bulk of the work's been done, it seems to be holding water quite well. So when you get to these high levels that go up and down, if there hasn't been something technically wrong, like the buffering capacity or some injection that's highly acidic or something like that that can throw off the measurement, we're probably looking at maybe temporal effects that are maybe unique to the situation you develop. Maybe it's somewhat contingent on the media conditions you have and things like this. Hopefully that helps answer that question. Did I get there?
Certainly, and it also partially already answers a follow-up question by Nick Jones. He also asked whether these other contributors to ECAR are cell-specific. And I mean, I assume the answer is also yes. I mean, a macrophage after activation makes itaconate, which is acidic. A T cell doesn't. So probably it also depends on the system you're looking at.
Exactly right. Now, because there is some buffering conditions that occur and hydration occur, we can control a little bit for that, those kinds of effects. But no, Ajit Divakaruni did a beautiful job when he stripped and took mitochondria out of the cell. And he was basically using proton production to look at ATP synthesis and catabolism, right? So you're right, depending on what you have in there. In fact, in the early days, GPCR activation extrudes a lot of protons through activation. We actually have some pharma people that look at basically ECAR rates as an indication of GPCR activation. So you're right, there can be other biological components, but those things happen very short. So the ones you're mentioning are usually fairly acute, not the macrophage one, but when you look over hours, 1 or 2 hours, the predominant flux that you're looking at in the way most people set up the experiments is, without a doubt, we know for sure is the anaerobic glycolytic pathway, without a doubt.
Good, thank you. When looking at this ECAR peak or this second ECAR peak after interferon gamma stimulation, actually I was wondering whether you looked at proliferation, and then there was a follow-up question by Daniel McVigar. It was a very nice paper also on the role of nitric oxide in rewiring glycolysis OCR, because in the end nitric oxide in mouse macrophages, it represses mitochondrial function. So is there changes along that side if you would plot the OCR, for example, or if you would do corrections for proliferation?
It's a great question. I unfortunately took too much of the data out to see the question he was asking. And that was really the Ed Pierce contribution, because when I saw that, I sent it to Ed. And you're absolutely right. The OCAR caves with the gamma interferon spike, and it's a nitric oxide. It's exactly what happens. So NO will inhibit respiration. So part of that glycolytic increase is probably compensating for that. But in the case of macrophage biology, that's the normal deal, right? It's not glycolytic many times compensating for dysfunction, right? And maintaining function. This is really what macrophages do, right? They live at this life-death valley very nicely. But yes, he's exactly correct. OCAR, if I showed you the OCAR, very nicely goes down with the gamma interferon peak in both cell types, as it turns out, which is very nice.
And then maybe the last one, there is a very interesting question from Kane Foster. If you consider now these CAR T cells in therapy, where do you think the advances will come from? Would it be from hyper-optimizing the metabolism to make them fit and generally increase their function? Or would it be more based on at the individual patient side and really look specifically within individual patients?
Yeah, there are two good open questions. We're doing a lot of work, and we're working with a lot of people. So something that we've been very much involved with. So we have at least we know the questions people are asking. Let's put it that way. So on the first part of the question is enforcing aerobic metabolism, the proof of concept. That's why I was very careful to say proof of concept. There's always the problem that then it won't go away, and you'll get all the adverse effects of the immune system. But we also said that when we were going to synthetically use lenti and other viral approaches, and so far there's been 10,000 patients, not a single transformation that's at least been noted at this point in time. So maybe there are other ways to get rid of highly aerobic cells that we just don't understand. So it may not be as dangerous as we think, and that's clearly what people are studying. However, that's why the arginine was seen as a very helpful thing, because the arginine, presumably the effects of conditioning of the cells would eventually go away, or it would be altered to the appropriate state in the right microenvironment once you debulk the tumor enough, right? But getting to your question about where is this leading? You know, everyone, we've been hearing now, it's because it's logical, it makes sense. Oh, we need an off-the-shelf product. We need something that we can treat everybody with, and that's allogeneic, right? And I can understand the allure of that, because now the problem is it's expensive. Manufacturing is hard. It's a time-honored task to do. It's individualized, which makes it logistically more complex, right? And so that was the attractiveness. But the reason autologous has gone so fast, who would have believed? And I did drug discovery in my early career. It was 10 years in, and now it's what, 15, and it's a couple $1 billion still. They've done it in 4 to 5 years, and it's going even faster because autologous is inherently a safer product. It's an easier product to work with. It's more likely, if it doesn't work, it'll be more likely just to be neutral as opposed to being negative. The minute you bring in an allo, you're bringing all the safety. And even though we've learned a lot, there's still a lot of safety, and it's not as straightforward. So allo may not be as straightforward off the shelf as people are thinking. So what some people are thinking is autologous, the idea of precision medicine, if it was fast, inexpensive, and we could do it very efficiently, like we do other individualized things, then why not stick with autologous? Because there's less to fix. There's less to go wrong in autologous. So I wonder, if you fix the logistical issues, does a lot of the need for these off the shelf go away, and we pursue a true green medicine, which is really utilizing and understanding the person's immune system and fixing it from that perspective. And so one of the biggest things we're seeing happening now is hospitals are building their own. I don't know if you saw, a couple days ago, MD Anderson bought Bellicum's 60,000 square foot cell manufacturing production. They're going to start making their own cars for their own patients. Sheba Medical Center did a beautiful job. They're using this new Lonza cocoon system, which is an amazingly closed system for cell production, and they have one of the best transfection technologies. And they're turning around cars to patients in a couple of days. There's no freezing and sending and shipping and all that, and very cost effective. And they treated 100 patients very successfully in a first trial. So will this become more decentralized? Is it appropriate that hospitals will do a better job with personalized medicine? Think about it. Pharma has a hard time doing that, right? Because they're trying to find drugs that can treat I don't think that'll go away. But for these life-threatening age-related diseases, maybe the personalized approach is the best. So there's a lot to do, but I don't think it's too early. It's too early to call. And then the last thing I'll say is what we said in our thing. Getting a cell to engraft back into a patient in the CAR T may be more a question of stemness. So if we solve the problem of getting a CAR to engraft and take residence and do well, maybe we really help the stem cell field, or vice versa. Maybe the stem cell field helps us understand better how to select and manufacture patient cells in a CAR approach to go back in.
Yeah, thanks for this insight from your side. This is, like you say, there is still a lot to do and obviously a lot to think about. And you also gave us a lot of insights for things to think about. So thanks again for your time, for your presentation, for your discussion. This was very useful for all of us. I think we will conclude here. So thanks a lot, David, for presenting. Thanks a lot for the audience for watching and providing this interesting discussion. Next week, we will have Annie Curtis. There were quite some questions the last weeks on circadian rhythm, so I'm sure you will like her talk. She leads the Clock Lab at our CSI in Dublin. So I also look forward to her talk. And I think meanwhile, you know where to find us on Twitter and on the internet. So I look forward to see you all next week. And thanks a lot, David, for your time. Bye all.
Very welcome.